Yeah, when I submitted that talk title, somebody said, it's a 20-minute talk, right? And they'll spend the first two just going through that talk title. So I actually put kind of a subtitle down here. And I really wanted to focus the talk in on how we're really trying to identify the right patients and the impact on their disease during the course of therapy. So I sit within Translational Oncology at AstraZeneca, and one of the cornerstones and the mission of this is to generate disease linkage data to guide patient selection in our clinical trials through the development of companion diagnostics. And then from gathering this data in the trials and the associated metadata, working this backwards and back translating it to really start understanding the tumor biology, the progression, the molecular progression of the tumor over time, and any acquired resistance in the tumor. So I want to focus on that last aspect, this acquired resistance to, this is a technology talk, actually. This is an information technology talk, right? So, but I want to have a backbone of a scientific story around it to share, so to kind of lend credence to the challenge that we have both in the translational oncology and then those in informatics and others that are trying to support this. So, for example here, I want to talk about our EGFR franchise at AstraZeneca. So it's pretty well understood that folks that have an activating mutation in EGFR respond really well to EGFR inhibitors. Sounds pretty straightforward. Unfortunately, we also know that the vast majority of them go on within about 10 months to become resistant to these inhibitors. And about 60% of this progression is due to a single amino acid change in exon 20 of EGFR, the T790M. So a really exciting drug in the portfolio of AstraZeneca is this AZD9291, which selectively binds and inhibits these T790M mutant patients. But invariably, several of these people go on to progress on this therapy. So one of the things that we've been doing within AstraZeneca is really focusing on, and it seems like many other people are too, gathering plasma samples from these patients, deeply sequencing them, and trying to understand these potential novel mechanisms of resistance. And this is the story that I'll weave, is what we're doing from an information technology data analysis standpoint to support these types of activities at AstraZeneca. So I don't think I have to beat this into the ground. This has gone over time and time again so far at this conference, this promise of the liquid biopsy. Some of the challenges of it are that the DNA is very fragmented, and there's very low copy number of the tumor cells within the plasma. So, we started experimenting, amplifying, the key, what we call the key AZ cancer genes, amplifying these up, and sequencing these out on PCR-based amplification platforms. And we were seeing really nice results here. And so, the price is dropping. This is no secret. The ability to cover areas of genes within these genomes is dramatically going up. The ultra-deep sequencing of, we used to think 30x was a lot, so, now we're in tens of thousands of X coverage. So we can really start thinking in terms about how we, select for the right patients to go into our trials, but also putting them on the right doses and the right schedules of these drugs. So as I mentioned, or maybe I didn't mention at the beginning, I head up the production informatics for AstraZeneca Oncology. And this here is our guiding principles. I think you have to understand the changing landscape, the dramatic increase in the amount of data, and also realize that there are still tons of artifacts in this data. But nevertheless, it's moving towards the clinic. So it's really important that we build tools that are not only flexible and scalable, but validated and secure. So this is what I'll use to lay out this technology story, as I mentioned to you. Processing our internal clinical samples quickly, but being able to change tools in and out very quickly as well. And it's great if you can adapt to the changing times, but if you don't arrive at the right answer, then it's not very useful. And then even after you've taken care of the first two, if you can't get it into the hands of the biologists and the clinicians, then it's kind of dead there. You need to empower them to be able to work through this data, share the data with the colleagues, and feel confident that they can do that securely. And and underneath all of this is a foundation of IT. So as I mentioned, and with this dramatic scale out of the number of samples now available to us through these liquid biopsies, and having to go very deep in sequencing of a lot of these, this proves to be a challenge to our current pipelines that we had in place and to the current IT infrastructure. So this is where we started working with Bina technologies to dramatically reduce this time to answer within our translational oncology. So, for example, we could take a relatively high coverage exome, which would take around 16 hours on our local infrastructure, and could reduce that to 45 minutes. And the same dramatic change in order of magnitude with whole genome sequencing. So this overall took the lab process from sample receipt to delivery of the answer back to the drug project team from around 10 to 6 days. The other thing that I think I want to emphasize is this is all done on commodity hardware. This is not locking yourselves into GPUs or FPGAs, and this is all done with open source software as well. So open source software. So you see the speed, right? It came. This is the 331 tools that we have released into production at AstraZeneca at this time. Yeah, I look at it every time. It scares me. So I can pause. I'll let you guys read that, and then we'll move on. This is one tool of the 331 that has over 50 command line options, right? So if you do that math, there's over 16,000 ways to get the answer wrong. And because most of the tools within our industry are academic tools and are constantly changing, you better have the ability to understand, have them well documented, have logging well done, provenance checking, and like we said, it's changing, so you've got to be flexible. So speed seemed like the easy part now when you start thinking about this insurmountable task. And there was a really interesting talk last night from the BioBuild folks, and we had come to the same conclusion internally that it takes anywhere between $100,000 and $200,000 a year of IT and informatics and engineering time to support this amount of tool sets, QCing these tool sets, validating these tools against, what I'll show you later, reference standards. So this is where we look to Bina to help augment some of the work that we were putting into this and eventually replace it. So we're asking them to validate the tools, upgrade the tools, keep them current, and make sure that we understand when we're processing the data, what's happened to the data, where it went, and all of these modular analyses that we need to do. All right, so let's assume we can cope with the scale that we need to happen in the often insane amount of time that people expect the data back in. How do we know what we're doing is correct? And then how do we fill those gaps when gold standard algorithms and other technologies fail us? So reference standards aren't new, but there's definitely been a resurgence of them over the past few years. I'll highlight one here, the genome in the bottle. It was started by Justin Zook and Mark Salad at NIST, and it's a great effort to establish an answer key to whole genome sequencing. But given that 90% of the sequencing done at AstraZeneca is for oncology, it falls short in several key areas. And it's not to say that it's not useful, it's just that it falls a bit short. And all of these things that I'm mentioning after talking with Mark and Justin, these are all on the roadmap for the genome in the bottle. But for right now, this isn't a cancer genome. It's not from FFPE or circulating tumor DNA. And at the beginning, it was mostly SNP focused. And as many of you know, most of the variants that we're interested in oncology, sometimes they're easy, most of the time they're not. So, but I will encourage everybody to go see Justin's new poster downstairs on how they're trying to tackle some of the larger insertions and deletions, and it's really moving a step forward. But we can really build off of these ideas around things like the ICGC Dream Challenge and the Global Alliance for Health, for Genomics and Health around their BRCA Challenge. These are two activities that are very interesting, and both AZ and our Bina colleagues have been actively participating in the Dream Challenge, and this is where people download data sets that are synthetic, have answer keys, and then you submit your data back. There's many people across the country, the MUTEC folks from Broad and others, that are actively participating in this. The one that's especially interesting to us at AstraZeneca is this BRCA challenge. So BRCA is an incredibly difficult gene to sequence, and so what they're doing here is they're focusing on not only the sequencing of this gene, but of the ability and the actionability of the variants that are found within this gene and how to prioritize those. But there's still gaps to be filled. So internally, what we've been doing is actually working a lot with the IDT, the Horizon reference standards. So these are known cell lines that are spiked together to give you known mutations at specific allele frequencies. And these are from FFPE, so although not truly the ancient FFPEs that most of us work with, we've seen some really nice results. So in fact, the mutation, the T790M that I talked about is spiked in at 1% in this mix. So we've been showing a lot of good success internally through our pipelines on validating those around these, both through hybrid capture and PCR-based amplification methodologies. And we've been extremely excited about the IDT pan-cancer panel that we've been running on this. We've been seeing a lot of really great results. But through some of the efforts of sequencing these reference standards, we've noticed that there are gaps within the current gold standard algorithms. And I say algorithms because nobody can ever, will never really settle on just one. But one of the things that we've noticed especially is complex variations in deep sequencing data. And the last thing that we wanted to do is develop yet another variant caller. But we developed yet another variant caller. So I'll introduce Vardict today. It's an open source piece of software. It's available via GitHub. I think this is a step out for big pharma that we're now producing and releasing and actively participating in open source and contributing this back to the community. But one of the real strengths of this is the complex variation in which it can tackle. So I think we all focus a lot on the calling of variants. And there's plenty of programs out there to help you prioritize and annotate these variants. But what about the areas that you can't get to, and why? And how do you know that you can't get to them? So one of the things that we're going to be doing with Bina over the course of this year is developing a highly scalable coverage database that will allow us to answer biological and technical questions across a host of capture technologies. And what we'll be doing is we'll actually be storing every sample that we've ever sequenced at AstraZeneca and will sequence going forward, coverage profiles of this with associated metadata from our LIMS system. So we've done it quickly. We can shake and bake, and we can do it flexibly. And for the most part, we're right. So now we need to get it in the hands of our scientists. So about a year ago, AstraZeneca started a program called Bioinformatics to the Bench, where we're really focused on providing interfaces into the data, helping people to prioritize the data. And again, this is something that's not very sexy, but often overlooked. But how do you simply share massive amounts of data with other people? So again, this is where we've leaned on Bina and their host of really great engineering teams and GUI experts to pull together these modules and GUIs to present to our end scientists. So there's two platforms, their RAVE platform, their analysis platform, and the AIM platform, which is their analysis and analytics around variant calling and hopefully soon to be expression and other data types. So this one I'm especially proud of because this really does show a true collaboration between AstraZeneca and Bina. So what we have been trying to do is actually work together to develop insights into the data that make sense from a cancer perspective. I think a lot of systems start with germline because it's the low-hanging fruit and then move to cancer, and we're actually doing that the other way here. So this is the co-mutation plot where you can look across different cancer types and different genes and really start seeing the mutation spectrum of your genes of interest. And then you can finally dig down into those genes and start seeing the mutation profile across the gene itself in what we like to call lollipop plots. So, but all of this isn't possible without some pretty cool IT technology, both on the databases side and everything else. Unfortunately, traditional IT isn't but refreshed every 2 to 5 years, and we're not only trying to solve problems that are very new today, but we're trying to design systems for problems that haven't even happened yet. So this is, no secret, this is where the cloud excels. AstraZeneca has built, over the past year, the AstraZeneca R&D private cloud, where we've launched things such as, high-performance computing clusters up there using Star Cluster and BC Bio. We've also recently integrated the Bina platforms into this. I think one thing that shouldn't go unnoticed is that there are a lot of platforms out there that are good cloud platforms. There's a lot of platforms that are out there that are really good local platforms. But to have to develop a local strategy and a cloud strategy not together sometimes is a little daunting. So this gives us this ability to process data in the same way, with the same algorithms and the same workflows and the same provenance, both locally and in the cloud. So, to summarize, let's do it. Let's do it quickly. Let's do it flexibly, but let's get it right. And yeah, that's all I have. So I'd like to thank the crew that really did all the work, both at AZ and Bina.